Dr. Jayanti Kunadasan, and I'm going to talk to you about a report about the 170 patients who changed the world, the patients who were the basis of the primary efficacy analysis of the Pfizer EUA. And I'm narrating this report on behalf of my co-authors, Ed Clark and Dr. Chris Flowers, as well as the fantastic data team and the other members of Team 3 of the War Room Daily Cloud Pfizer document volunteers. Now, we have to remember that the EUA was granted on how well the drug was shown to have worked. Now, that is the efficacy. There are other endpoints that were looked at in the trial: the reactogenicity, the safety of the drug, and the immunogenicity as to the titers that were produced on because of the drug. But the EUA was granted on the basis of how well the drug prevented one getting the COVID infection, and that is the efficacy. So there are many efficacy endpoints, but the EUA was granted on the primary efficacy endpoint. Those who got an infection after 7 days of the second dose with no evidence of an infection prior to this. And I'll explain this a bit more clearly after this. Now, the planned analysis for this was to be done when they got 164 patients who fulfilled the criteria. However, they got 170 patients, hence the topic that we have today. Now, if you think about it, these 170 patients who we have in the final analysis have to have no protocol deviations, because by definition, if you were found not to have followed the protocol, you can't make it into the trial population in which we are analyzing the efficaciousness or the effectiveness of a drug. So this is the EUA authorization, and this is what I mean by the 170 population. So final analysis of efficacy against confirmed COVID infection from 7 days after dose 2 in participants without evidence of prior SARS-CoV infection. And this was basically the evaluable population. So they had to check which population in this trial would make it to be evaluated to be able to enter the efficacy analysis. And what they found were there were 162 patients in the placebo group and very few patients in the vaccinated group who got infected. So they said that the vaccine had a 95% efficaciousness, and that this essentially was the basis of the EUA. Who qualified to be part of this population? So the evaluable efficacy population, you had to have no evidence serologically or virologically of having a COVID infection prior to 7 days after dose 2. You had to be symptomatic. So if you had at least one dose or one symptom of fever, increased cough, increased shortness of breath, chills, muscle pain, and then now that you're presenting for an illness visit, we would do a swab, and this illness visit could be an unplanned visit because you don't know when you're going to get sick. So at that point, you would have symptoms as well as a positive nasal swab at a central laboratory or an acceptable local test. And then you would qualify to be part of this population of evaluable efficacy. And how are they going to analyze this population? So because they were planning to do interim analysis throughout the trial, they planned to do interim analysis when they had 32 cases that fulfilled this criteria, or 62. So they couldn't do the 32, so they did interim analysis at 62 patients, 92, 120. And when they reach 164 patients, and if they had statistical analysis, they would say that they would have fulfilled the criteria of efficacy for this trial. So now, looking for these 170 patients was difficult. So where did we find them? So we found them by compiling a list from 2 tables, was basically the interim lab tables, and they were buried in page 586 out of 671 of this document and page 66 of page 217 of this document. And you look at 16.2.8.2, what you see is that subjects with the first COVID occurrence from 7 days after dose 2 and without evidence of infection prior to 7 days after dose 2. So this is the evaluable efficacy population. And we've seen those words before. So you had to have your dose present for testing an unplanned test illness visit more than 7 days after dose 2. You had to previously be seen to have no evidence of infection of COVID. So that's what this means. Visit and binding assay and the serological and virological evidence of no infection. And then you had your symptoms. So that's why they listed symptoms. And this is the test result of the positive test as well. So this is the test result of the positive test of the symptoms that you had acceptably 7 days after dose 2. These patients, these 170 patients, were also found in the narratives document. So in the narratives document from page 1059 to page 2506, there's a section where they narrate everything. So you can find the age, demographic data, and pretty much the entire patient record for that one particular patient. So the reason that they took a lot of trouble to actually compile this is because this is the most important patient population in the trial. These are the patients on which the EUA were granted on. So again, you have evaluable population, but they mixed it up. So you have to look under the categories of evaluable and or severe. So severe would be those who got severe COVID. So you have to sieve out the patients who probably got COVID, severe COVID, even after dose 1. So even when you look in the narratives document for the 170 patients, you have to do a little bit of cross-checking and making sure that you take out the patients who just got 1 dose or who only got severe COVID but didn't qualify to be in the 170 patient. So we found the 170, we compiled the list. The list is in the Excel spreadsheet that we have put in the publicly available Daily Cloud website at the end of our report. So we found these patients, and what we decided to do is we cross-check the list that we got with publicly available demographic data. So that's what we're trying to show here. We've publicly cross-checked our patient list with the age groups, the sex, and which countries they're from, as well as the race, as well as whether they were obese or not. So we're pretty confident that we found the correct list of patients. Now, what did we find when we first found them? So the first thing, we have to have another look at the EUA. So this is the document from the Emergency Use Authorization. Basically, it winnows down the populations, and the trial started with 43,000 patients. We ended up with somewhere around 37,000 patients. But look at the exclusion criteria. Those who excluded are those who did not meet eligibility criteria. That's pretty, self-explanatory. Did not provide informed consent and did not provide all vaccinations as randomized or did not receive dose 2 within the predefined window. And here they've put the predefined window as 19 to 42 days. So that's interesting, because if we look at the trial protocol, what we see is that the trial protocol, and we have to remember, and I did not include it in this slide deck, but it is explained quite clearly in our Daily Cloud report number 41. This Pfizer trial initially started because this is a new drug. Initially, we didn't know what dose we were going to give, and we didn't know how many times we were going to give that dose, and we didn't know the dosing interval. So initially, the trial was, did we give just one dose, or if we give two doses, is it 21 days apart or 60 days apart? Those were the windows that they considered. So that's why the word window comes in, because we actually had two windows. We were choosing between a 21-day window or a 60-day dosing interval window between dose 1 and dose 2. Protocol Amendment 5, and this is the last protocol amendment before the trial for phase 3 properly started on the 27th of July, 2020. They've very clearly stated, if you look at Protocol Amendment 5, and it's explained quite clearly in the Daily Cloud report, that we've decided on the dose 30 micrograms, 2-dose regimen, 21 days apart. But in the protocol, having allowance for human behavior, the window seemingly is determined to be 19 to 23 days after visit 1. And also what is written in many parts of the protocol is visit 2, vaccination 2 is 19 to 23 days after visit 1. So this is what is explicitly written in the protocol throughout all the amendments from Protocol Amendment 5. And Protocol Amendment 5, as I said, is the protocol amendment where they decided what dose they were giving and at what time interval. At any point in the protocol after this, you only find the words 19 to 23 days after visit 1, and essentially that we have settled on a 21-day dosing interval. And if you look at it, we have analysis sets because you need to know what you're going to analyze. And pretty much the analysis sets, this was again in protocol in the Protocol Amendment 5, that what we had was dose 1 immunogenicity data. We're going to determine it 21 days after dose 1. But the evaluable efficacy, and remember, we've seen those words before. Those were the words that we saw in the narrative section. So this is, again, the primary endpoint of the trial. So all eligible randomized participants who receive vaccination as randomized within the predefined window, having the efficacy measurement after the last dose of the study intervention and determined by the clinician to have no protocol deviations. So the evaluable efficacy, what we can see for dose 1, we can write that we want 21 days, but in the evaluable efficacy of this trial drug after a 2-dose protocol, we're not willing to put the words predefined window. But seemingly in our protocol, again, one would think logically that the window that we want is 19 to 23 days, because that's the window they've actually stated. And does it matter if we've changed the dosing protocol from 19 to 23 days, as is explicitly written in the protocol, to 19 to 42 days? What we have shown, and we are going to analyze the entire patient data, but we've done a quick analysis from the 44,000 patients down to 37,000 patients. What we find is when you change the dosing interval from 19 to 23 to 19 to 42 days, we can recover an extra 14,000 patients. And of those 14,000 patients, it includes 5 patients from our very important final efficacy analysis population. Now, I'll take a bit more time to explain this graph. Remember, this is the number of patients enrolled in the trial. Some of them were eliminated because they didn't receive the dose. Some only received dose 1. And then this is finally the number of patients who received 2 doses, dose 1 and dose 2. Now, remember, the final efficacy analysis included patients who have had COVID 7 days after dose 2. The trial cutoff date is November 14th. No one vaccinated on the 8th of November and after that could actually qualify to be in this population because they would not have reached 7 days by the time November 14th arrived. And November 14th is the data cutoff date, and after they were evaluating patients for the EUA. Now, there are some patients who were vaccinated days 18 and below. They were eliminated, obviously, because they are protocol deviations. And the patients who are vaccinated after the data cutoff date, and they are eliminated for these purposes because they couldn't have been evaluated for the EUA. Now, let's look at the number of patients differentiated. If we take the protocol as it is written, 23 days plus, obviously, this is the 7 days after dose 2. What we have is 1,500 patients. But if we actually took 42 days interval between dose 1 and dose 2, we would only be eliminating 171 patients and not 1,581 patients, as we can see the difference between these two. So essentially what we're able to do is we're able to recapture 1,410 patients, and importantly, 5 patients from our 170 population are in this dosing interval. So we have to ask the questions, what is the proper method of introducing a protocol amendment? And why are we accepting, in effect, a doubling of the dosing interval from a 42-day dosing interval between dose 1 and dose 2, and the 21-day dosing interval that was decided in the trial protocol? Those who are vaccinated 18 days apart are considered protocol deviations. Those who are vaccinated 41 days apart are not. But those who are vaccinated 43 days apart could be now protocol deviations too. But we have to remember what is explicitly stated in the protocol is 19 to 23 days. The 19 to 42 days only appeared in the EUA. And it was only probably appeared by being deliberately ambiguous in the statistical sets by actually just stating the words predefined window without actually putting a number next to it. We have to look at the statistical analysis plans. So remember, the statistical analysis sets in the protocol documents don't have a number. But the statistical analysis plans is it's a plan of how they're actually going to analyze the data. So the statistical analysis plan for phase 1, 2, and 3 was finalized on November 2nd, and we only get to see version 3. We don't get to see version 1 and 2. And chronologically, it doesn't make sense because the statistical analysis plan for phase 1 of the study, when they're doing the dose finding and etc., was finalized on the 18th of November. Implicitly, the study design still says it's a 2-dose to a 21-day apart, 30 microgram Pfizer BioNTech study. But for the first time in the analysis sets, and if you compare the analysis sets found in the protocol document with the analysis sets found in the statistical analysis plan, there's a number. But remember, this was finalized on the 2nd of November, and this is version 3. We can't see version 1 and 2. The dose 1 immunogenicity for phase 1 is the visit window is basically 21 days. That's what they mean by 19 to 23. And remember, what we are looking at is the evaluable efficacy. That's what you see in the narratives. The dose 2 is defined within a predefined window of 19 to 42 days. So even in the statistical analysis sets, it's a 2-dose separated by 21-day schedule. But what we're analyzing is patients within a 19 to 42 days dosing interval. And so that's a protocol deviation, because we have to ask ourselves, how do you correctly actually introduce a protocol amendment? What is the proper method? Do you actually explicitly state it in the protocol, or do you do it in this manner in which they have seemingly done by having deliberate ambiguity in the analysis sets and only introducing a number at the end of it? And this looks, instead of a predefined window, it looks like a post hoc defined window, because this is defined somewhere in November. Now, remember, the 170 population had to be perfect, because you would have taken patients who didn't follow the protocol out of this population. So let's look at some other examples, and you can do this by yourself when you have the list of the 170 patients, and you can actually put this number into the abstracted tool on the daily cloud, and all these documents will appear, but I'll take you through them. So this is one of our patients who made it to the 170 final efficacy analysis population. Remember, this is the population that on which the basis of the EUA is. So these patients, 16.2.8.2 in that table. So subjects occurrence, first COVID occurrence from 7 days after dose 2 without evidence of infection prior to 7 days. So back to this. So this patient had dose 2, had symptoms 42 days after dose 2, and prior to that had no evidence of infection, as evidenced by negative, then had symptoms of fever, and then had a positive test. So this patient made it into the 170 population. So you can see them in table 16.2.8.2, as well as the narrative section where you have the word evaluable again. And you see 1068, 1062, and you can have a little bit of the narratives about this patient, but also see which list that this patient made it to. Listing of important protocol deviations. So if you go into this document and you look up at the beginning of where this deviation subcategory is actually possibly did not receive correct dose of investigational drug. So this comes to the absurdity of what we're actually looking at, that this patient who made it into the final efficacy analysis population, the 170, possibly did not receive the correct dose of the investigational drug. This is another patient that we've highlighted. So back, this patient's made it to our table 16.2.8.2. There you find them there, received their dose 2, had symptoms and had no evidence of infection, then had fever 21 days after dose 2, and had a positive test, and so has COVID. Made it to our narrative section as well under evaluable. And there's a little bit of a narrative about this patient, but this patient also made it into this list, which they really shouldn't have. This is a listing of important protocol deviations, phase 2 to 3 analysis, and received blood of plasma products immunoglobulin 60 days before enrollment, and that is actually protocol deviation number 15. So this patient really is a strong protocol deviation and shouldn't make it into the list. Now, the threshold, if you remember from the chart earlier, to be able to perform a final analysis was 164 patients. What we have is 7 patients who were outside the dosing window of 19 to 23 that was explicitly stated in multiple versions of the protocol throughout, plus 2 patients with hard protocol deviations. So now our number from 173 comes down to 170 comes down to 163 patients. What we're saying is we don't even qualify to do a final analysis in the first place. Thus, the basis of the EUA needs to be reconsidered. Other things we are also highlighting, and you can find in the report, are other patients who were withdrawn prior to the issuance of the EUA, one of the patients who was withdrew because they were upset about their clinical management in the trial. So this raises ethical questions. And if you consider the number of patients who are withdrawn and add it to our patients that we're saying already have protocol deviations, the number further goes down to 161. And some interesting patients, so this patient 10161004, again, add this into abstractor and you will find the relevant documents. Had symptoms 2 days after dose 2. So that would have disqualified them because remember you had to have no evidence of infection prior to 7 days after dose 2. Now, this patient had a swab that was for symptoms 2 days after the in dose 2, then that swab was only processed 18 days after that episode, and then it came back negative. And then he presented for illness again sometime later and had a positive swab. But if the patient had actually had a positive swab 2 days after dose 2, that patient couldn't have been part of the 170 analysis. And that's other questions that we want to raise as well. And the other issue is that we've had a lot of sequential numbers. So now, remember, this is 170 patients out of a population of about 37,000 patients, each with an equal chance of being in the final efficacy analysis for this trial. So we welcome seasoned statisticians telling us the probability of having even one set of sequential numbers. But what we found is 6 sets of sequential numbers appearing in the 170 population. We think that raises a lot of questions and we welcome debate from other eminent authorities. Thank you very much. I hope I've made that clear. Thank you.
It's Naomi Wolf of Daily Clout, and I am asking you to please, if you like the video you just saw, support us, become a member, donate. You can send checks to P.O. Box 24, Millerton, New York, 12546, or go to Daily Clout, DAILYCLOUT, become a member or donate. Thank you so much for your support. Every penny goes for paying our hardworking staff, paying hosting costs, and paying our lawyers who have been leading the fight to keep you safe and free, to keep the Constitution safe and to keep you free. Thank you so much.